Ligia Watanabe, a postdoctoral researcher at the University of São Paulo, Brazil. I would like to start my presentation with this question. What if the higher selenium intake does not reflect on the blood selenium adequacy? Before going to the title that is a spoiler of my presentation, some facts about selenium are relevant. First of all, selenium is an essential nutritional trace element. The relationship between selenium level and health effects, represented by a U-shaped curve, suggests illnesses associated with both deficiency and excess. Several endogenous conditions may impact blood selenium levels, but our focus was to identify genetic variants associated with serum selenium deficiency in Brazilian descendants. We performed a genome-wide association study to identify single nucleotide polymorphisms, or SNPs, potentially associated with selenium deficiency, and I will present all steps with the corresponding results. We started collecting data and biological material from 382 patients. The characterization of our sample indicated variations in age, body mass index, and the predominance of female in the sample. These characteristics were included as covariates in the association analysis. We also observed serum selenium deficiency and adequate selenium intake. After DNA collection, we performed the genotyping array, identifying 640,000 SNPs. Then, our packets were applied to quality control and imputation. After these steps, 90,937 SNPs remained. We used a case control design to perform an extended version of a traditional mixed model for GWAS, and the maximal statistical analysis to avoid the genetic model misspecification. In the first analysis, we found 57 SNPs in the dominant model, 67 in additive model, and 32 in the recessive model. The maximum statistical analysis identified 162 SNPs. Among them, 44 SNPs were in common with the other genetic models. The post-GWAS analysis filtered 12 SNPs. Here, we highlighted 2 SNPs and revealed the title of the present study. GWAS identifies genetic variants indicating a role of impaired thyroid metabolism in selenium deficiency. The 2 SNPs were previously associated with impaired thyroid metabolism, and the prior studies suggested a contribution of thyroid disturbance to serum selenium deficiency by reducing selenium and selenoprotein's availability. In conclusion, this GWAS reported the association between selenium deficiency and SNPs in genes not directly linked to selenium metabolism, reinforcing the complex relationship between selenium intake and the factors that affect the selenium requirements for optimal health.